Hello everyone, we are the Futures. Today my teammate Steven and I are here to give a presentation on our team project, Kidney Savior.
According to Lancet, the most authoritative journal in the medical area, there were 697.5 million cases of chronic kidney disease in 2017. Nearly one-third of those patients lived in two countries, China with about 132 million cases, and India with about 150 million. Further, 10 other nations, the US, Indonesia, Russia, Japan, Brazil, Pakistan, Mexico, Nigeria, Bangladesh, and Vietnam, each had more than 10 million cases in 2017. As a Chinese student, I was so stupefied to realize the truth that 1 in every 10 Chinese is suffering from CKD, whereas I had never heard of this disease before. So, by far I think many of you are surprised by the facts in the previous slide and may wonder, what is chronic kidney disease? The definition and classification of chronic kidney disease, CKD, have evolved over time, but current international guidelines define this condition as decreased kidney function, shown by glomerular filtration rate, GFR, of less than 60 ml per minute per 1.73 square meter, or markers of kidney damage, or both, of at least 3 months duration, regardless of the underlying cause. That is to say, kidney function gradually declines over time. And CKD has 3 main features which are good to be known about. First, it is mainly caused by diabetes and hypertension, which are quite common in recent years. Second, since there are no obvious symptoms at the beginning of the illness, it is called invisible disease. Third, people with CKD are 5 to 10 times more likely to die prematurely than they are to progress to end-stage kidney disease. And end-stage kidney disease, which requires dialysis treatment, is also highly fatal if not properly treated. Here are the 4 conundrums faced by CKD patients. The first one is increased risk of death. CKD may cause other diseases such as uremia and cardiovascular disease, and even can progress to kidney failure. The risk of death rises exponentially as kidney function worsens and is largely attributable to death from cardiovascular disease. Globally, chronic kidney disease directly resulted in an estimated 1.23 million deaths in 2017, with an additional 1.36 million deaths attributable to cardiovascular disease resulting from impaired kidney function. The second one is lower quality of life. Health-related quality of life is substantially lower for people with CKD than for the general population, and falls as GFR declines. CKD has so many symptoms, including nausea, vomiting, tiredness, loss of appetite, sleeping difficulties, persistent itching, and so on. The third one is limited chance to get treated. In 2016, more than 1 million uremic patients in China needed dialysis each year. However, the treatment rate of patients with end-stage kidney disease is only 50%. The final one is high cost of treatment. A research by a group of graduating medical students from the University of Guiana found that over a 5-year period, the total cost for a patient with dialysis is roughly $57,000 compared to $184,000, which is the cost of maintenance treatment for transplant patients. To illustrate the importance of our technical advance, let us see a comparison with traditional methods of treatment. For the traditional methods, one of them, dialysis, has certain unavoidable shortcomings. The first is that it is repeatable, which means patients need it continuously even 3 times weekly. Also, it takes 3 to 4 hours to have one dialysis, so it is quite time-consuming. Besides, in the process of dialysis, people will surely feel pretty painful and have a high risk of infection. For another traditional method, kidney transplantation again has some weaknesses, such as high cost, which will put heavy financial burdens to the patient's families. Also, since the demand of kidney is often higher than it can be supplied, it is hard to find kidneys, not to mention gaining a matching one. By contrast, for our new method, it must be of lower cost compared with transplantation and safer compared with dialysis. What's more, it is more radical, since it prevents the production of bad chemicals instead of filtering it after its production. Moreover, we can also ensure its enough supply, Therefore, from the above comparison, we can safely see the significance of our advance. I guess now you may think our new technology, Kidney Savior, sounds like so excellent and wonder whether it is possible to achieve. The answer is yes, and our secret of our new technology is probiotics. Probiotics are live bacteria and yeasts that are good for you, especially your digestive system. We usually think of these as germs that cause diseases, but your body is full of bacteria, both good and bad. Probiotics are often called good or helpful bacteria because they can help your gut healthy. You can find probiotics in supplements and some foods, like yogurt. Doctors often suggest them to help with digestive problems. Paracresol, which is a significant component of a chemical substance that may bring to kidney diseases, is able to be dealt with a new substance. This new substance is polyphenol oxidase, a kind of engineered probiotic.
Now we'll talk about the goals and what we aim to achieve through our project. Our main goal is to relieve the pressure that CKD patients face by slowing down the progression of the disease. We know that P-cresol is a precursor of PCS, a key toxin that's harmful to kidney health. Thus, our team aimed to construct an engineered probiotic that can degrade P-cresol, hence prevent the synthesis and accumulation of PCS. Therefore, our engineered probiotic can be beneficial for CKD patients. So how do we achieve our project goals? We will now present an overview on our methodology and our experimental procedures. In order to achieve our project goals, we designed several parts to construct our engineered strains. Firstly, some necessary basic parts were designed, including a coding sequence of the core protein PPO, P14 promoter that worked in our probiotic NISO 1917, a transcription factor TYRR, TYRP promoter regulated by TYRR, and ribosome binding site. Based on the above basic parts, we designed 3 composite parts, working for expressing PPO protein and BL21, testing the tyrosine-induced system, and applying the tyrosine-induced system to express PPO protein in the probiotic NISO 1917 respectively. We first constructed 3 plasmids and transformed them into E. coli competent cells, respectively, through molecular biology technologies. Then, we cultured the E. coli BL21 DE3 with number 2 induced by IPTG and purified PPO protein. Finally, we tested the function of the 3 aspects. We cultured the engineered bacteria with plasma number 1 induced under different concentrations of tyrosine and detected green fluorescent values to test the function of the inducible system, detected the enzyme activity of PPO purified protein, and fermented the engineered bacteria with plasmid number 3 to test the function of P-cresol degradation. Now moving on to the results of our experiment. We mainly did experiments and data analysis in 3 aspects. First, the construction and functional verification of tyrosine-induced promoter. Second, the purification and activity determination of P-cresol degrading enzyme. And third, the degradation activity of P-cresol in the engineered bacteria induced by tyrosine. First, we will talk about the construction and functional verification of tyrosine-induced promoter. We knew from the reference paper experiments that only in the presence of tyrosine does the tyrosine promoter start and break down P-cresol. We constructed the inducible system on the PSU 2718 P15A vector, which was used to initiate the expression of GFP protein firstly, to detect whether the inducible system of tyrosine worked normally. The constructed plasmids were transformed into E. coli NISO 1997, or ECN, receptive state of intestinal probiotics, and the recombinant strains were obtained by adding different concentration of tyrosine. As shown in Figure 1, with a low concentration of tyrosine, the induction system responded well to the tyrosine concentration in E. coli NISO 1917. However, as shown by Figure 2, when the concentration reaches 1,000 micromoles, the fluorescence intensity of the strain decreases, so the high concentration of amino acids may have an inhibitory effect on the strain. It suggests that we should use 50 to 500 micromoles of concentrations of tyrosine to induce the PPO protein expression. Second, we will talk about the purification and activity determination of P-cresol degrading enzyme. We first extracted a large amount of PPO proteins from the modified E. coli BL21DE3. PPO enzyme solution was purified for further confirmation by the SCS Page method, which is found in the corresponding protein strip. Then, we measured the enzyme activity of PPO protein by analyzing Figure 4 and defining the enzyme activity of the PPO protein. We learned that the enzyme activity of PPO is about 0.5 U. Through the determination of purified in vitro enzyme activity of PPO, we found that PPO in E. coli can have very good expression and excellent catalytic properties. The analog of P-cresol can be successfully oxidized and metabolized to produce colored products, which can be further used in the construction of engineered strains. Third, we will talk about the degradation activity of P-cresol in the engineered bacteria induced by tyrosine. We first exposed the modified E. coli to different concentrations of tyrosine to express the PPO protein, and we used the PPO proteins to degrade catechol, which is an analog of P-cresol. It was found that the yellow color got darker gradually with the increase of tyrosine concentration, indicating that more P-cresol was degraded. We further obtained the PPO crude enzyme solution by ultrasonic decomposition from samples with different tyrosine concentrations, and then detected the PPO enzyme activity with catechol as a substrate. It was found that the enzymatic activity of the crude enzyme solution on catechol increased significantly with the increase of the concentration of tyrosine, as shown by Figure 6. This shows that tyrosine could indeed induce the expression of active P-cresol degrading enzyme. All in all, our experiment results were in line with our expectations in the three aspects.
We list three objectives of our modeling here. Firstly, we want to give you a general impression about data we used for modeling. Here we come to the visualized description about the interrelationship among OD600 values, inducer concentration, and reaction time. We make this multiple fitting through presenting such a response surface, whose fitting effect is quite good. Here are the detailed fitting effects of the response surface. We choose the reaction time of 2.5 hours to explore the relationship between OD600 values and inducer concentration. The R-square is quite good and proves the equation's good fitting for forecasting the relationship between inducer concentration and OD600. To forecast the relationship between OD600 and reaction time at fixed inducer concentration, we use the data circled in blue frame. Here comes our modern results. A linear equation reflecting the relationship between OD600 and reaction time when the inducer concentration is 100 micromole.
In order to understand local communities' knowledge about CKD, we gave out questionnaires to the public and received results from 274 participants, ranging from 15 years old to 60 years old. After personal information, we start with a straightforward question, asking them whether they have ever heard or learned about CKD. Over about 84% of them either have never heard of it or they don't know it in details. Also, participants were asked to answer what would they prefer to do if problems appeared to their kidney. Based on this question, consulting a doctor was the overwhelming choice, about 87%, and taking medicine ranked the second at about 49%. What's more, we questioned whether they would take in probiotics if they were beneficial for their health. Gladly, most of the answers turned out to be a yes. This gave us great confidence in our product because it proved that our product would definitely have a position in the market with an established customer base. So, according to the results we got from the questionnaires, we took some actions to improve our project. Firstly, we decided to spend more time and energy on educating people about CKD because the public's low awareness on it. We hold an educational lecture at Xuhui Health Center to educate the locals about chronic kidney diseases, including its causes and some current treatments, along with the social impacts of our research project. To attract more people, we designed mugs, hats, and canvas bags with our logo and sold them to the public as a form of propaganda. The cost of hosting offline promotion activities and making the products were all covered by our online fundraising, about 4,000 RMB in total. We also created a group chat for all of our participants, so that we could keep them informed and interested in our project. The lecture finally became a complete success. Over 80% of the participants showed their interest in our future steps and were willing to share our project to their friends and relatives. Secondly, to gain more expertise on our project, we interviewed Dr. Gao Huayang from the Chinese Academy of Sciences and Dr. Ting Xunzu, a researcher in Gansu Center of Microalgae Engineering Technology. These interviews made us realize that we have neglected the presence of a strong competitor, healthcare products. According to professors, these products have already established a firm base in the market, so it would be a challenge for us to persuade people to purchase our products, as they would prefer to try these healthcare products more. To address this issue, we decided to invest more in promoting our products, including selling our self-designed derivative products and publishing more articles on our WeChat subscription account. Here are some articles from our WeChat subscription account, broadcasting our project topic, relieving chronic kidney disease, by utilizing engineered probiotics and our experiment progress. In order to get a more comprehensive research project and increase our impact of influence, we collaborated with the following two other IGEM teams. Neptuner aims to use engineered probiotics to reduce the negative effects that result from staying up late. They kindly give us suggestions about our business plan as well as human practices. To be more specific, they helped us to better position our product in the market. We also posted each other's derivative products on our online stores and WeChat subscription accounts to further boost sales. Atro Extinct contains excellent high school students all over the world. They use a synthetic biology technique aimed at converting atrazine into cyanuric acid, and in the presence of atrazine in certain samples, the fluorescent protein appears green to the naked eye, making it easier to detect. Atro Extinct joined our educational lecture at Xuhui Health Center and also gave us many useful advice about our whole project design and demonstration. After briefly introducing both teams' projects, we asked questions and for each other to further improve project design.
Now I will briefly go over our safety considerations when during our experiments. Safety concerns were carefully followed during the entire experimenting process. Despite the regular laboratory safety that is followed for all experiments, we also had safety procedures that are specific to our own experiments. Biosafety cabinets were required for crucial steps in our experiment. The biosafety cabinet can only be used when it was functioning normally. We also made sure that the glass baffle in the biosafety cabinet was closed during use, for the ultraviolet rays are harmful for human eyes. Other special equipment include homogenizers, shakers, and ultrasonic processors. We use plastic containers with such equipment because glass can break and release infectious material that can harm the operator. When using a homogenizer, shaker, and ultrasonic processor, the equipment was covered with a strong and transparent plastic box and disinfected after use. And moving on, now I will discuss what are some other things that can be done in the future about our project. Although our results conform to our expectation, there are some things that can be improved in the future. There are two ways to make PPO protein get in touch with P-cresol more effectively. The first way is to overexpress the transporters of P-cresol so that the PPO protein can get in touch with P-cresol and degrade them more effectively. The other way is to fuse a membrane protein with PPO so that the PPO protein can be located at the surface of the cell membrane, allowing to degrade P-cresol more efficiently. Furthermore, considering that the intestinal tract is in an environment with low oxygen concentration, we can express anaerobic degradation gene of P-cresol, which comes from other microorganisms, so that we can further improve the ability of degrading P-cresol in anaerobic environments, such as the intestinal tract.
We are the filters. Filter out harmful substances from human body and create a pathway to a better life. Thank you for your listening.